Unfortunately, we are all susceptible to diseases and disorders that disrupt the human body's natural and normal functions and cause uncomfortable symptoms and even death. However, we are all also born with natural healing entities called embryonic stem cells that can be used to treat a multitude of diseases and even heal wounds. As we grow into adulthood, these embryonic stem cells mature and lay dormant in bone marrow tissues and other tissues. As you will see, the use of these cells in medicine and engineering is vital to the future of healing and eradication of disease. Stem cells were discovered in the 1980s when scientists in a lab found specialized cells in rats that could be re-entered into the cell cycle and re-specialized into any desired cell. Advances in human stem cell research did not really begin to take off until the beginning of the 21st century. This is when mesenchymal stem cells, referred to as MSCs, came into focus. MSCs had the same characteristics as those discovered in the lab rat cells. That is, they could be derived from specialized cells, such as bone marrow cells, re-entered into the cell cycle, and converted into desired cells for healing damage and replacing ones that had died. As stated before, stem cells have the ability to heal a multitude of diseases and disorders. They do this through two important properties. Differentiation is the ability of a stem cell to turn into the desired cell after re-entering the cell cycle. Proliferation is the ability of the cell to reproduce and grow in quantity. These factors are solely dependent upon the stem cell's microenvironment, which is why research on this topic is so important. Without a healthy and stable microenvironment in which to grow, these cells will ultimately not differentiate or proliferate, and this will lead to the death of the cells and an inability to heal. This brings us to the importance of researching and refining the constituents of stem cell microenvironments. One of the 14 engineering grand challenges of the 21st century is to engineer better medicines. Stem cells are the ultimate medicine because they do not merely treat the symptoms of disease, they have the capacity to cure them entirely. A problem with stem cells, as other cell or tissue transplantations, is a shortage of donors. There are tens of thousands of people that await transplants each year, and the amount of donors cannot possibly supply the demand. Scientists have recently proposed use of stem cells to alleviate this problem. If healthy cells can be grown in a laboratory or in vitro, in a flask or petri dish, there would be no need for donors anymore, thus solving the supply versus demand problem. Not only would stem cells be able to treat diseases, they could be used to engineer and regenerate tissues by growing livers, kidneys, hearts, and so on in the lab. In order to do this, the scientists have to make sure that the microenvironment in which the cells grow have close to perfect conditions to assure proliferation and prevent mutations or cell death. The constituents of a cell's microenvironment include soluble factors such as growth factors and hormones. Growth factors function as the name suggests. They help the cells quantify. Hormones like estrogen are vital for growing osteocytes or bone cells. Some hormones, like the parathyroid hormone-related protein, affects MSC differentiation, which prevents them from proliferating. This is why these soluble factors must be controlled in a lab to make sure the beneficial ones are provided and the detrimental ones are eliminated or left out. More important factors of stem cell growth and proliferation are substrates and extracellular matrices. In the lab, these are synthetic components on which to place stem cells for them to proliferate, which make them abiotic. Bioceramics are man-made substrates on which to place stem cells to help them reproduce quickly and effectively. Their material and space size are vital for growth and depend on the type and size of the cell being grown. This picture of a scanning microscope image illustrates an experiment where rats' osteogenic cells were placed on nylon sieves with different textures and space sizes. S0 in the upper left-hand corner was a flat surface where the cells did not differentiate quickly or efficiently, causing their yield to be very low. However, the scientists found that the closer the space size got to the size of the rat cells, the more efficiently and quickly the cells proliferated. Quadrant S3 in the bottom right-hand corner illustrates the texture and space size that yielded the most amount of differentiated cells. For the future of regenerative medicine and tissue engineering, a group of scientists has proposed a machine called a bioreactor, pictured on the right. It would simulate the most stable microenvironment discussed before and cause stem cells in the lab to differentiate and proliferate in greater quantities at higher rates. This would make growing new tissues and organs in the lab a reality, where they could be used for sick or dying patients that currently wait on a list for treatment. The bioreactor is proposed to be made of low-cost, readily available materials, so the manufactured organs and tissues would be much cheaper than ones provided by a donor. The current cost of donor tissues is exorbitant because the demand far surpasses the supply. Engineering new organs would eliminate the need for donors by creating a higher supply, thus lowering the cost. In the next slide, I have input a cartoon video that perfectly wraps up the subject of engineered tissues using stem cells. This video is optional but very interesting, so thank you for watching.
Regenerative medicine. Regenerative medicine is a therapeutic treatment that replaces or regenerates diseased or damaged cells, tissues, and organs in the human body to restore normal function. The treatment can be by injection of small molecule drugs, biologics, medical devices, biomaterials, and stem cells, or alternately involve growing tissues in the lab and then implanting them into the body. We asked people, what excites you, what frightens you, and how will it change the way you live? These were some of their responses.
Of course this sounds great, but will regenerative medicine like this really be available in 2020, or is it just hype?
Medical researchers say that recent advances in stem cell technology and the ability to create brand new tissues from cells in the lab have brought us much closer to seeing this treatment a reality. Some treatments are already in clinical trials. For example, using human embryonic stem cells to repair the effects of macular degeneration in the eye. This means that in the near future, stem cells injected into the body could be used as a treatment for everything from wound healing after accidents, right through to the specific treatment of childhood diseases such as type 1 diabetes, or the repair of cardiovascular system after heart attacks and the repair of damaged brain tissue caused by Parkinson's, stroke, and other brain injuries. Doctors will be able to regrow brand new organs from stem cells in the lab, ready for implantation into the patient. This sort of treatment will open the door for the widespread replacement of damaged organs with healthy new ones without the need for donors.
But where will these cells come from?
That will depend on the type of disease or disorder that will need treating. Certain types of stem cells may be better for particular ailments. There are 3 types of stem cells: embryonic stem cells from early stage human embryos, adult stem cells derived from our body cells, including the baby's cord blood, and pluripotent cells generated in the laboratory from our body cells to behave in a similar way to human embryonic stem cells. Current research uses all types of stem cells. Some people have raised questions regarding the ethical implications involved with stem cell technology. This is because ethical concerns have been raised over the use of human embryos for embryonic stem cells in regenerative medicine. Research using human embryos is undertaken under strict guidelines established through legislation and approved by university ethics committees.
How will this technology impact on an aging population?
Often, technologies don't always turn out as we expect, and there may be unintended consequences even from technology with good intentions. Regenerative medicine may be the first step to us living longer, healthier lives, but will this just mean we have an even larger, more dependent, aging population? What do you